Hello, and welcome to Let's Meet the Virologists, a podcast about the people behind today's science headlines. People just like you, working to understand viruses and how they affect you. During the COVID-19 pandemic, we are talking with postdoctoral researchers involved in coronavirus and COVID-19 related research so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. Today, August 27th, 2020, we have with us Dr. Shabali Das, a postdoc in the Catter Lab at Washington University School of Medicine. Shabali got her PhD in biochemistry at the Bose Institute, affiliated with the University of Calcutta in India, studying immune-mediated aspects of tuberculosis disease and vaccines. She has continued this work during her postdoctoral training at WashU and recently had a first author paper in Nature identifying a protective role for specific innate immune cells with the title Group 3 Innate Lymphoid Cells Mediate Early Protective Immunity Against Tuberculosis. Hi Shabali, happy to have you with us today. Tell us a little bit about yourself. How did you become interested in microbiology research?
So I was born and brought up in India. And then India is endemic for so many diseases, mostly the infectious diseases like tuberculosis, leishmaniasis, cholera, and so on. When I was planning to do my higher studies, I was really interested to learn more about the various infectious disease process. And then I wanted to contribute to the society by making some sort of vaccines or drug that will help the people. So, and then I was so keen to study in depth all these, at least some of these infectious disease process. And then for this reason, I have done my bachelor's and master's degree in microbiology back in India. Then I did my PhD in tuberculosis. So I have a long connection with tuberculosis.
Thinking back, how did you become interested in research? Do you have family members that are scientists?
No, actually, in my family, there is no one who's working in a scientific lab or who are scientists. But I used to get that interest in microbiology from my school when we used to have those practical classes where we used to get to know the microscope and all these tiny fields. So from then I have decided, I have to do something with this tiny world, which is not visible, but they have so much impact in our everyday life, from making yogurt to killing people. Everything is associated with microbiology.
In the environment that you grew up in, were there diseases caused by viruses as well?
Yeah, actually, there are so many viral infections as well, like chikungunya. And then sometimes there are some seasonal flus as well, swine flu, bird flu. So we grew up with all these viruses and bacteria all around us.
In a little bit more detail, can you tell us about the steps you took to get where you are in your career today?
So once we passed our school, then we had to decide what path we want to choose. So I thought I will study microbiology, and then I ended up attending the college in my city itself. So I did my bachelor degree there, and then after doing my bachelor degree, I wanted to learn more about this thing. So I did my master's degree in microbiology. And at that point, after doing my master's degree, I was very sure that I'm gonna pursue research as my career goal, because the thing which we lacked in our school, we get to know the microscope or some tiny bit of the microbiology world, but in our bachelor study and master studies, we had to do a lot of practical implication based on those microbiological approaches. So then I thought, okay, I would like to do a PhD to get to know this field more, and then if I can contribute to the society either by contributing to the basic science field or in the application field.
How did you start your postdoctoral studies?
So I wanted to get more training, and then like my planning was to get more training from USA, and then I only applied to USA. I have not applied to Europe or any other places because from like Dr. Khade's lab or some other like TB labs, we have seen so many incredible papers which are like the base of the tuberculosis research. So from my like PhD times, I used to follow all these labs, and then I applied to Dr. Khader, and then she was very excited to like have me as a lab member in her lab too. So yeah, I think my journey so far is incredibly exciting, and then all the supports from my mentors, my fellow colleagues, and of course my friends and family, they helped me kind of where I am today.
Was it difficult to move to another country?
Yeah, so yeah, it was not easy. I mean, it was really hard because we have like cultural differences, we have language differences, everything. But the thing what connect us is the science.
Can you describe in a little bit more detail the work that you've been doing in the Cotter Lab? So pre-COVID, what kind of work were you doing?
So, as you know, Khader Lab is like a TB lab here. There are like 3, 4 TB lab in Washu, and we are one of them. So we study various aspects of tuberculosis, disease, and then the control process in mouse model. So I am basically interested in the vaccine efficacy, and then like vaccine delivery, vaccine formulation, this part of the tuberculosis study. I also did some work on the early mechanisms that either lead to like disease progression or control in the mouse model.
Can you describe a little bit more the vaccine work? Obviously, for TB, looking for a new and better vaccine has been a long-term goal. What are you working on?
In our lab, we are testing different formulations, different routes, as well as we are like testing the hypothesis why the current vaccine, that is like BCG, is not effective, and how we can make the same BCG vaccine effective.
So can you describe to us what was the most exciting eureka moment that you've had so far in your career?
Actually, last year we published one of our study in Nature, and so I think for me that was the eureka moment for me so far. We got like so many responses from the field. Everyone is very excited about that paper.
Can you describe that research a little bit, that paper?
In that study, we have identified a very rare cell population in the mouse model, and then we have seen the similar cell population in non-human primate and human model. As I have mentioned you, we have like multiple collaborators all over the world, so they helped us like to tease apart the mechanism, how these tiny, like very rare cell populations are important. And this was the first time we were like kind of talking about that cell type in the tuberculosis disease mechanism, and then that was associated with the early control of the disease.
So sort of conversely, what is the most difficult thing you've had to overcome as a scientist? And I guess, how did you overcome it?
Yeah, so that difficult moment was also associated with this paper. So when we got comments back from the reviewer, they wanted us to do like 1,000 other experiments to prove that that cell type is true, and then the effect is true, all these things. So you know like how nature works. So that cell type was so rare, and then I had to do some intracellular staining to like detect that cell population. And then that intracellular staining was like so sticky, and then it was binding to like some other background cells or something. So it took me almost 4 months to standardize that staining technique. And then sometimes I used to get frustrated I mean, even I cried because I'm like trying so much and it's not working. And then thanks to Dr. Khader, she like kept me motivated all the time. And then finally, like after doing various kinds of like standardization, we finally get to like identify that cells.
What about personally? If you think back to your undergrad or master's or graduate degree, what was the most difficult thing you've had to deal with or overcome?
Oh, so you know what? Like from my childhood, I never stayed out, like stayed without my parents or without my family. I have done all my studies, like in even like my PhD. I have done everything from like my home because all my institutes were like 10-15 minutes driving distance from my home. So I used to stay in my home and then I did all my studies. So then I decided, okay, I have to like go out of my comfort zone and then I have to go outside. So the initial decision was very hard. And then like when I was in my final year of my PhD, so I started applying outside and then I got one offer from UPenn. And then after I got that offer, I decided, no, I don't want to like leave my country, or I don't want to leave my parents. So I will stay in my country and then I'll work here. But then later I decided, no, if I like bind myself with all these things, then I cannot like reach the point where I really want to get to. Otherwise, all my studies, all these days, is not gonna be that effective as I envision it to be. So then I started applying to like other labs and everything. And then when the final like day was there, when I have to leave my country, I was so sad and I even like cried multiple times. So I think that was like really hard for me. That decision was really hard.
So you've been very successful in your postdoc. How did you overcome the initial reluctance to leave behind your family, your support system back home?
Yeah, actually, Dr. Khader was like very supportive from the beginning. She used to have a conversation with me regarding my family and everything. She wanted to like make me feel homely here. And then luckily, I have got like some good friends here. I made like friends here. And then I used to like call my family like every day, twice or thrice.
Your career has just started. Where do you see yourself going from here?
So I want to like be an independent researcher, and then I want to establish my own lab and then work on the TB field.
So if you had a chance to ask your older self, so you say in your 60s or 70s, so this is after most of your career is finished, what would be one question that you would ask?
I would ask how my contribution to this science helps this society. I think that's the question I asked from the beginning of my career. That is still question to myself, like at present, how my work is contributing to the society. And then that would be the question to my total self as well.
The Catter Lab is not known as a coronavirus lab. How did you start working on COVID-19 related research?
Yeah, so I mean, as a scientist, like everyone wants to like contribute to the COVID research in one way or the other. One of our collaborator, Dr. Deepak Kaushal in Texas Biomed, he started working with the COVID-19 infection in the non-human primate model. And then he is a very good collaborator with Dr. Khader. And then Dr. Khader and Dr. Kaushal, they wanted to study the immune responses in the non-human primate model, which will be like close relative to the human infections. So, so then that kind of like started our journey to the COVID field, that event. And then, for the COVID research, the most important things are BSL-3 facility and then the BSL-3 training. So luckily we had both. We have people who are like well trained with in the BSL-3 environment. And then Dr. Khader provided me the opportunity to work on that COVID uh, research. And then I'm really excited to get that opportunity.
Follow up on that, can you tell us a little bit more about the research that you're doing on COVID?
So Dr. Kaushal, he's in the primate institute. So he like had access to various monkey species as well as like different age group. So he could like do a lot of like infection thing. And then he sent us the samples like the bronchoalveolar lavage or lung tissue samples and everything. And then the another important thing is that we have a like dedicated single cell RNA sequencing core in our BSL-3. So it was really helpful. So initially we proceed with the single cell sequencing on the monkey bile samples, and then we have included like various time points and then various age of the monkey, all those things. So I think it will really help us to map the immune network or like immune components that are associated with the disease progression, or what are the changes that are like permanent in the monkey bile, all those things, all those information we'll get from this.
So this was following up on your previous work on tuberculosis, in a way applying it to COVID.
Yeah, and then once we get to know those like cell population which are like protective, so we can use this strategy or use this knowledge in the future vaccine development as well.
So on a more personal level, how has the COVID-19 pandemic affected you and your family?
So like everyone else, it impacted me a lot. Firstly, when we initially heard about the ramp down, so that actually delayed all my like upcoming experiments for a couple months, I would say. And then you can imagine I'm like in fifth year of my postdoc. So now I have to apply for like some career development grants and everything. And then I wanted to get some preliminary data for that thing as well. So everything is just like delayed for a couple months. And then the other thing is that like when we used to go to lab, we could interact with our like fellow lab mates and then fellow friends. That was really a booster for me. I mean, we could talk about science or non-scientific things and all those things. So that was like completely gone. And then finally, I think due to this COVID thing, since my family is in India, and then I'm not able to make any travel plan to India for this COVID situation.
To follow up on that, as a microbiologist, how do you make decisions about how to keep yourself, your family, your coworkers, and your community safe?
I always like use my sense, like where should I go or what should I do or what I should not do. So I always like think before I do anything, even if I have to like go to grocery. So I think, is it really necessary or can I order it online? So I usually tell the same thing to my friends and of course my family. So, and then I always keep reminding them like wear the mask properly, because lots of people wear mask, but that's not the proper way of like using it. Even the mask, like the quality of the mask, that also important. So you have to like think about everything. And then I like ask them every time to like wash their hands frequently and then use the sanitizers and all those things. I mean, they have been like listening to this thing like from various sources, but when it comes from me, they are like more like willing to listen. And then the social distancing is very important. So I think if like from my side, if I do my part of job like properly, then that will motivate like the people around me. They could be like my friends, my family, and as a whole we can keep the community safe in the long run.
So you've had some downtime in the past couple of months. Have you started any new hobbies during this time?
Yeah, actually, so during this COVID time, we had some free time, right? So I registered for some online courses and then some webinars. So I get to see through some more talks than before, because previously we had like some due to work schedule and everything, we were not able to like be in some of the important talks what I really would like to attend, but due to my like work schedule I could not. The other thing is that I set up a fish tank during the pandemic because I was feeling like so lonely, and then I wanted to have someone with me. So that's why I set up a fish tank so I could take care of them during my free time.
You were talking about how you are still communicating with your family. How do they view sort of the COVID crisis in India, but then also how do they think about what's going on here?
Yeah, actually, you know what? Like the situation currently in USA is worse. We all know. But the Indian media is showing that in a way that everyone in USA is like died, is they're like dead. So my parents they're like very scared that I am living here and then I have to go to lab and all those things. So I have not told them that I'm working on COVID samples. I did not tell them, and then I would not tell them. So that's the thing. And then in India, our social systems and then our like economic system, everything is different in there as compared to here. And then, you know, like there are a lot of people in there. So even it's not possible for them to keep a 6 feet distance between like two people. So my parents are like sort of they're staying inside the house as much as possible. And then I am ordering like some stuff for them from online because they are not that familiar with the online purchasing and all those things. And even they don't want to buy things online because they don't trust the online thing there. So yeah, it's been hard, but thanks to God, everyone from my family is safe so far.
I guess, so we're sort of wrapping up our podcast. Any last messages for our listeners? I guess any thoughts about the future of the COVID-19 pandemic here and in India? Where do you hope that things are going?
Yeah, so I do have a message, and then that would be please act sensibly and then do respect the safety of others. I mean, if you are not wearing mask, you are not only risking yourself, but you are also risking the people around you. So it's very important to respect others' safety, I guess. And then I am hopeful that we will have a vaccine soon, and then that will end the pandemic. I am hopeful to see a COVID-free life like we had before.
Thanks, Shabali, for talking with us today about yourself and your research. Shabali's previous work with TB has allowed her and her collaborators to quickly begin studies to identify the immune cell components associated with protection or disease severity following SARS-CoV-2 infection to ultimately design better vaccines. This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google and Apple Podcasts or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologists@gmail.com.